Now coming to the bacterial growth curve in NEET 2018 question was asked from bacterial growth curve. It is very important. What is the correct sequence of the bacterial growth curve? First of all, there is a lag phase. Now they can even ask a sequence based question from the bacterial growth curve. What is the correct sequence of a bacterial growth curve? It is lag phase. Then we have log phase, which is also known as exponential phase. Then we have the stationary phase, and then we have the death phase or the decline phase. So this is the correct sequence of the bacterial growth curve. So first of all, there is a lag phase. Then we have a log phase, which is also known as exponential phase. Then we have a stationary phase, and another one is the death phase, which is also known as decline phase. So these are the phases for a uniform staining reaction or a morphological study. Bacteria should be stained in a logarithmic phase, which is a log phase. Why in the log phase? Because in the log phase, there is increase in the number of the bacteria. So starting from the lag phase, there is accumulation of the metabolites, and there is increase in the size of the bacteria in case of lag phase. So size is increased in case of a lag phase. Now coming to the log phase, in which number of the bacteria is increased. Since we have a lot of bacteria, the maximum number of bacteria is present in the log phase, which is also known as exponential phase. Now, since the population of the bacteria is more, therefore you can easily see bacteria in the microscope. So that is the reason that we do staining, we do morphological study in this log phase, which is logarithmic phase, which is also known as an exponential phase. Any of the term, if it is given, this is log phase. Very important log phase, because the maximum multiplication occurs in the exponential phase. So basically population doubles in every 20 minutes in this log phase, and here the increase in the number is so much that it is to the power raised to the n. This is increase in the number in case of an exponential phase or the log phase. Now coming to the stationary phase, as the name suggests, growth of the bacteria is going to be stationary. That means the viable bacteria, viable word means that the living, that means the bacteria that are able to germinate, or the living bacteria is equal to the dead bacteria that doesn't grow, or that doesn't divide, or that doesn't form the colonies in the solid medium. So viable bacteria is equal to non-viable bacteria. Bacteria are present, there is no growth, there is no multiplication. So viable bacteria is equal to non-viable bacteria in the stationary phase. Sporulation, the endospore formation happens in this stationary phase. So another important point is sporulation also happens in this stationary phase. So in case of a harsh environment, basically it is the survival mechanism of the bacteria. So the coping mechanism to prevent themselves by forming the spores, because spores are resistant to heat. What part of the spore is resistant to heat? So it is the dipicholinic acid, which is present in the core of the spore, which is resistant to heat. And because of the harsh environment, because of the inability to replicate, it's a survival mechanism that occurs in the stationary phase for a bacteria by forming the spores. Now the death phase, which is also known as the decline phase, the bacteria die faster than they multiply. So there is accumulation of the toxic metabolites, and this results in the death of the bacteria. So the question that is asked from this bacterial growth curve, either they can ask a sequence based question. So first a lag phase, then log phase, which is also known as exponential phase, then the next one is the stationary phase, and then the death or the decline phase. They can either ask a sequence based question, or they can ask there is exponential increase in the number during which phase. This is a very frequently asked question. Increase in the number is seen in case of the log phase, which is also known as logarithmic phase, which is also known as exponential phase. Rapid division of the microorganism occur in which phase? It is again occur in the log phase, which is also known as exponential phase. So generation time, the interval of time between the two cell divisions, or the time that is required for a bacteria to give rise to two daughter cells under optimal condition, is known as the generation time, or the population doubling time. Since one gives rise to two, the population is doubled. So for many medically important bacteria, the generation time is 20 minutes. The total count is the total number of cells that are present in a sample. Either they can be viable or non-viable. That means viable means either they are able to multiply, or non-viable means they are not able to multiply. So either they are living or not. Total count means the total number of the cells that are present in a sample. While on the other hand, if we say viable count, that means the live cells that are present. So these are the total number of cells that are capable of multiplication. So this is a method, viable count is a method which is widely used in the water bacteriology for the estimation of this coliform count. So coliform count is used for the drinking water. So in filtration of the sewage water, we do the presumptive coliform count in the drinking water. And this is a test that we have talked about in the public health also.